Hello everyone, and welcome back to our fourth and final module of Chapter 3 on Biological Psychology. So as promised, this one is going to be a lot shorter. It's 10 slides in total, and that includes a couple of intro slides and things like that. So hopefully this one won't take us very long to get through, and this will be your class for next Friday, the 30th of October. So once I get this one posted, the whole all of next week's lectures will be uploaded. So that'll all be done in advance. So we'll get started. The fourth module is essentially about measuring and observing brain activity. So what we're going to be talking about here are some ways in which we can intervene in the brain and then observe what happens as a result of that, but also ways in which we can observe and intervene in behavior or cognitions and then observe what happens in the brain during that process. So in terms of mapping the brains, for years, two different approaches have been used to study the brain. The first was to study patients who had already had some brain damage or have had a part of their brain removed due to a disease or injury. And the other is to stimulate brain areas of patients who are having brain surgery. So those are really the two only kind of natural ways we'd be able to observe this type of relationship. However, obviously these methods rely on accidents to create an unhealthy brain. So you're not just able to intervene in somebody's brain in these ways unless there's kind of a medical reason to do so. So these days there are two broad approaches used to currently map the brain. The first is to do something to temporarily affect certain brain areas and then observe the consequences that has on behavior. And the second, as I mentioned earlier, is to manipulate behavior and then record the effects on the brain. So, in terms of intervening in the brain and observing behavior, there are less of these methods than of the types in which we intervene in behavior and observe the effect on the brain. The first method would be the lesion method, which is a technique in which researchers intentionally damage an area in the brain. So a lesion is essentially an abnormal or damaged brain tissue. So obviously we can't do that to human participants for ethical reasons. It has been done sometimes in animals, and in some rare cases it is done in humans for a medical purpose, like we just discussed, but we can't just take a bunch of healthy people and kind of damage certain areas of the brain to see what happens. So we have two other ways in which we can intervene in the brain temporarily. So the first would be transcranial magnetic stimulation, or TMS, which creates a virtual lesion. A wire coil is placed on the person's head, and essentially it delivers an electromagnetic pulse to a targeted area of the brain. So we're using magnets, which is why it's called transcranial magnetic stimulation. And what that pulse does is it causes the neurons to fire. So it can be used to briefly inactivate an area of the brain and then observe the effects that it has on a particular behavior. The second method is an even newer method known as transcranial direct current stimulation. So in this one, we're not just using magnets, we're actually using an electrical current. So it's applying a very small electric current to an area of the cortex, and the direction of the current basically dictates whether the activity is going to be suppressed in that area or stimulated. So for example, different directions of the current might increase memory or they might inhibit performance, depending on what area you're activating or deactivating at that time. So again, all of these are ways in which we can do something to the brain to affect its functioning, and then we're able to observe what effect that has on behavior and cognitions. On the other hand, we can also intervene in behavior, so we can get people to perform a certain task or ask them to think of a certain thing, and then observe what effect it has on the brain. So essentially, there's kind of a field called structural neural imaging, which is a type of brain scanning that produces images of the different structures of the brain. And this can be accomplished using a bunch of different types of technology. So electrodes, which you've probably all heard of, may be used to detect electrical energy of neurons in particular brain regions. They could be connected to a machine that essentially translates them into energy. And so typically electrodes are hooked up to a machine like an electroencephalogram, which is the next thing here on the slide, which makes a brainwave recording produced by a whole bunch of electrons that are attached to the scalp. So it's useful in that we can see whether there's activation in general in the brain and in kind of the general area, but it is not very precise, so it can't really help us narrow down exactly what areas of the brain are being activated during a particular task. Then we have magnetoencephalography, or MEG, which is a neuroimaging technique that measures the tiny magnetic fields created by the electrical activity of nerve cells in the brain. So when we were talking about something called TMS in the last slide, basically that is using a magnetic force in order to activate or deactivate areas of the brain. In this case, we're actually measuring the magnetic fields that exist in the brain. And so what that tells us is what areas of the brain are active at that particular time. So because it's measuring the fields created by the electrical activity of nerve cells, we're able to kind of target what areas of the brain might be activated during a particular task. And then we also have positron emission tomography, or PET scans, PET scans, which is a type of scan in which a low level of a radioactive isotope is injected into our blood, and its movement towards regions of the brain during a particular task are measured. So very similarly to the MEGs, what you're trying to do is you're trying to locate exactly what area is being the most active during a certain task or during a certain type of kind of cognitive activity. And so basically what's occurring here is that we know that the areas of the brain that are using the most energy, that are the most active, are using more energy, and so therefore there'll be more blood flowing to those areas of the brain because they're going to want to be using up more oxygen. So when we see this radioactive isotope that's in the bloodstream, if it kind of concentrates in a particular area of the brain, that indicates to us that that brain area is particularly active at that time. So next we have computerized tomography, or CT scans, which you've probably heard of. So it's a structural neural imaging technique in which x-rays are sent through the brain by a tube that rotates around the head. So again, just like with the EEGs, this one isn't super precise, but it can be very useful in cases when magnetic or electrical interference are discouraged. So for example, if somebody was in a really bad car accident and you didn't know if there may be some metal shrapnel or something embedded in their skull, you wouldn't want to be using magnets in order to examine them, right? Because that could obviously move the metal material if it did exist within the brain and could really cause some major damage. So CT scans are often used as kind of a preliminary step in kind of trying to see generally what's going on within the brain and if there may be any kind of structural abnormalities. Magnetic resonance imaging, or MRIs, which I'm sure you've all heard of, use powerful magnetic fields as well as radio frequencies to take highly detailed pictures of the brain. So it's essentially based on how neural regions absorb and release energy within a magnetic field. So again, it's basically looking at where those fields are most active, which would kind of indicate to us where the most activity is occurring at that point. So this type of MRI is known as a structural MRI. But there's also what's known as a functional MRI, or an fMRI, which can see brain activity associated with specific thoughts or behaviors. So it's one of the most precise methods. And so basically the receivers detect the influx of oxygen-rich blood into specific brain areas that were just active. So again, just like in the PET scan, you're looking at which areas are most active by examining where the blood flow was most concentrated, because of course, again, the most active areas are going to be using the most oxygen and therefore they're going to have a lot of blood flowing to them. So the active areas would then be producing bigger signals, and it allows us to kind of localize brain activity in that way. So it's not just kind of observing what's going on or kind of looking at the structures within the brain, it's actually helping us to localize that brain activity to a specific area. So here are just some kind of pictures from your text of some of the different types of structural neural imaging. So you can see in the first figure here in 3.33, we have a CT scan in A, we have an MRI scan in B, and we have a diffusion tensor imaging scan, which we didn't really cover, but it's another type of structural neural imaging technique. And then in figure 3.36 over here, you see functional magnetic resonance imaging, or fMRI scan. So you can see how the typical MRI scan in the other figure, like 3.33, you can see that it kind of shows you the structure of the brain, but doesn't really indicate specifically what's going on. Whereas in the fMRI in 3.36, you can see that it's actually able to kind of localize that brain activity to a particular area. So some things to keep in mind when mapping the brain is that technology cannot replace critical thinking. So sometimes brain images can convey oversimplified and misleading impressions, and they can make small contrasts look dramatic. as well as in some cases they may mask significant variability. So depending on how they're presented and how much of an explanation is given, if you show somebody, you know, a picture, for example, like that diffusion tensor imaging picture where there's all those colors and everything looks like it's kind of really active, without actually explaining what's going on there, that can cause people to kind of draw their own interpretations. So we really need to be careful when using these types of techniques to present our findings in a way that kind of really explains what it is we're looking at. So unfortunately, a lot of the time, researchers will include brain scans in their articles because they seem more scientific, but that's not always the case. fMRI research sometimes uses poor statistical procedures, so they use what we call inflated correlations, and so again, that's going to possibly make results look like they are a lot more dramatic than they are. There's a really interesting example of how results can be misinterpreted. That is shown by a group of scientists who conducted an fMRI on a dead salmon, and it actually showed activity. And so basically what we kind of learned from that is that even though it's seeming like there's activity in a certain area, you really need to have a baseline for comparison, because sometimes things are going to react even when there's not actually any activity occurring. So you need to kind of have a picture of what a typical, you know, brain would look like, and then be able to compare it to what's going on in a particular circumstance, instead of just looking at it and saying, oh, well, it looks like there's activity there, so something must be going on. So you kind of need to use a control in a way. And so what we're seeing from all these trends is that the real problem is not in the technology itself, but it lies in bad theories, poorly defined measures, and inappropriate interpretations. So it's unlikely to reduce the complex behaviors to single brain locations. It's very unlikely that we'll ever be able to kind of fully understand what's going on in the brain, but these techniques can help us in a lot of ways to kind of see generally where brain activity is occurring and generally what parts of the brain might be associated with what functions. So that is it for this module. This is the official end of chapter 3. I hope you enjoyed it. As I mentioned, I know that it's a lot, but if you can grasp kind of the major details, like the bits about neuron structure and communication and the different divisions of the nervous system and some of the major brain areas and what they do, like that's really what we're hoping that you take home from this chapter. So don't worry about memorizing everything, but hopefully you enjoyed learning a little bit more about you know, how these factors can influence our behavior and our thoughts. So I will end there, and I will, I guess, see you in the next video when we start chapter 5 on consciousness.